Okay, hello students. I hope the sound is audible. Okay, hello students. Okay, so we had discussed the initial seven chapters. Now we will be discussing the eighth chapter, self-variant of life. So we discussing some important diagrams and the diagram related questions. Not just diagram related questions, but we'll also discuss the concepts as well related to these diagrams and specific tables. Okay. So, when we are discussing with respect to the eighth chapter, that is cell unit of life, here we will be discussing about some of the cell size, okay, and the smallest cell, the largest cell, largest single isolated cell being the egg of ostrich, okay, and these are the lemon cells, attractive, okay, and eosinophil cell. This is our nerve cell, the longest cell, okay, columnar epithelial cells, WBCs, and the RBCs, okay. Now, we will also discuss some few points about eukaryotic cell. Eukaryotic cell, as you can see, it is having nucleus. Okay, the nucleus is covered by nuclear membrane. Along with the nuclear membrane, there is presence of the ER as well. That is endoplasmic reticulum. If they are having ribosomes, they are called as RER. If the ribosomes are absent, they are called as SER. We also find the mitochondria, the Golgi apparatus as well. Okay, so they have given some information here. A typical bacterial cell is nearly 1 to 2 micrometer. PPLO, that is puro-ammonia-like organisms, which we commonly call it as mycoplasma, the smallest cell. Okay, PPLO is around zero point 0.1 micrometer. Okay, then viruses, they are smaller than the bacteria. Okay, in biological classification, it is clearly mentioned that bacteria is what 1 to 2 micrometer, but there are some bacterial filters. Okay, those bacterial filters were passed by some other organisms. Those organisms were only the viruses. Okay, so remember the name DBS from biological classification. Okay, so Dimitri University. Okay, WB Stanley. Okay, and we have GE cases. That's Stanley and University. These were the three people who were involved in explaining the structure. First, Dimitri University was responsible for saying that there are some organisms which are smaller than viruses. Okay, and the person who actually said that the virus, the name, the term was then given. Then later on, the viruses were present in the form of crystals. Okay, so this was later discovered that the viruses outside the host body, they are in the crystalline form. Okay, so remember these three things. Next, so this is plant cell and animal cell. Now, plant cell, as you know, the majority of the space in the plant cell is occupied by vacuole. The outermost layer of the plant cell is cell wall. Next to cell wall, there is presence of cell membrane. Now, here in between these cell walls, you can see some space. This space is itself called as a plasmodesmata. Now, that's a very important labeling here, plasmodesmata. What is actually plasmodesmata? Is the connection between the two plant cells, adjacent plant cells. It acts as a bridge. wherein the cytoplasmic exchange takes place between one cell and other cell with the help of plasmodesmata. Okay. Then the other structures that are present in a plant cell but absent in animal cell are chloroplast. Chloroplast is only seen in case of plant cell, not animal cell. Not only that, here lysosomes are usually absent in plant cell, whereas lysosomes are present in animal cells. Okay. Now, apart from that, the outermost covering of a animal cell is plasma membrane, which we also call it as cell membrane. Here, the outermost covering is cell wall. Usually, the vacuoles, if present in animal cell, they are very small in size. Usually, they will be absent. Okay. So, centrioles are again present in animal cell but are absent in plant cell. Okay. The major difference between plant cell and animal cell. Next, this is a structure of fluid mosaic model of plasma membrane. Okay. The fluid mosaic model was given by Singer and Nicholson in the year 1972. Okay. So if you can see here clearly, here there is a bilipid layer structure. Okay. The head part is hydrophilic, whereas the tail part is hydrophobic. So the head part usually comes in contact with water, whereas the tail part is not in a direct contact with water. If it comes in contact with water, the structure will collapse. Okay. So to avoid that, the tails are usually present internally and the external surface, the head is in contact with the external surface. Now, the gaps between these particular head and tail is filled by cholesterol, and usually the lipid, this is what we call lipid bilayer. If at all there is some lipid, the fat or some lipids if they are present, they can easily pass through the plasma membrane. But what if it is a water? As we said, the tails are hydrophobic, means they hate water. If they come in contact with water, the structure will not be the same. So, how the water and water soluble components will enter the cell and how they can pass out from the cell, that's mainly because of presence of the proteins. Okay, there are two types of proteins: extrinsic protein and intrinsic protein. Extrinsic proteins are the proteins which are present externally. Okay, they are present externally on the surface outside the membrane, and if they are embedded, means if they are present in the plasma membrane, they are referred as internal or integral proteins. Okay, that are very essential for transport of water and water soluble proteins, not just proteins, are the substances that are transported from one cell to another cell. Now, there is also presence of some glycoproteins. Okay, what is mean of glycoproteins? Glyco means sugar. If those sugar molecules, if they are in contact, these are the sugar molecules. They are in contact with the proteins. If the sugar molecules, if they are in contact with the proteins and they are forming a structure, that is what we call glycoproteins. Now, if the sugar molecules, if they are in contact with the lipids, if they are directly in contact with the lipids, they are called as glycolipid. Now, both the glycolipid and the glycoprotein, they are produced from Golgi apparatus. Remember a process called as glycosylation, wherein the glycolipids as well as glycoproteins they are being produced. Okay. Now, also remember the function of plasma membrane. It is it does not allow all the particles to pass through them. It is selectively permeable in nature. If it is selectively permeable in nature, it allows only the selective molecules to pass through the membrane. Okay. Now, it acts as a covering, outermost covering in case of animal cell, whereas in case of plant cell, it is the second most covering. Okay. Not only that, the transport here, if we talk about the transport, there are two types of transport that we observe with respect to molecules. One is passive transport, and the other is active transport. Passive transport refers to the movement of molecules without the expenditure of energy. Now, without expending the energy, if the molecules are moving across the membrane, then this is called as passive transport. The example for passive transport is osmosis and diffusion. And for active transport, the the movement of molecules from either from external cell, I mean the adjacent cell to inside the cell, or the molecules which are present inside the cell, but if you want to go outside the cell or to adjacent cell, if they are moving against the concentration gradient. For example, if they are moving from the lower concentration to higher concentration, if the movement is happening from lower concentration to higher concentration, the energy is needed. Okay, so with the expenditure of energy, if the process is taking place, it is called as the active transport. Usually, the example for active transport is sodium potassium pump, which we observe in case of the neural system, which we'll be discussing. Okay, how actually the generation and conduction of nerve impulse we'll be discussing that. And not only this, even during the ATP formation, ATPase. So during all this process, we discuss about active and passive transport. Next, so this is the structure of endoplasmic reticulum. If you see, the outer membrane of nucleus is itself forming the coiled structures. So that is what is going to form the endoplasmic reticulum. If the endoplasmic reticulum, on top of endoplasmic reticulum, if you see the ribosomes, then such type of layer is called as ARIA, rough endoplasmic reticulum. If on certain endoplasmic reticulum, if you don't see ribosomes, absence of ribosomes refers to smooth endoplasmic reticulum. RER helps produce the proteins, helps synthesize the proteins because of presence of ribosome. The main function of ribosome is production of proteins as it is a protein factory. Smooth endoplasmic reticulum means here the ribosomes are completely absent, and here we see the synthesis of steroids, okay, lipids and steroids, steroidal compounds. Now, with respect to Golgi apparatus, you have to remember that there is a cis phase and trans phase. The proteins that are formed from the ribosomes present on ER or the ribosomes that are present in the cytoplasm, those proteins will enter the cis phase and comes out from trans phase. Remember, okay, the matured or the modified protein will come from trans phase. The immediate formed proteins they enter through the cis phase where the proteins get manufactured, I mean modified, and then the modified protein will come from the trans phase. Okay, a very important feature of Golgi apparatus is packaging and modifying of the proteins according to the requirement of the cell. Not only that, just we discussed that Golgi apparatus is a very important cell organelle for enzyme synthesis of glycoproteins and glycolipids, which we call glycosylation. Okay, then the next one here you can see the ribosomes. There is a larger subunit and smaller subunit. Larger subunit in case of prokaryotes. Larger subunit in case of prokaryotes is 50S, and smaller subunit in case of prokaryotes is 30S. So it becomes 70S. And in eukaryotes, The larger subunit is 60 years and the smaller subunit is 40 years and the combined form is 80 years. So remember 50 plus 30 not 80 not like that. Here there is segmentation that is taking place because of the overlapping of larger subunit with smaller subunit. It is going to form 70 years in case of prokaryotes and the largest subunit is 60 years in case of eukaryotes and 40 years in case of the smaller subunit. Both when combined, when the sedimentation was observed, it was found to be ATS. ATS in case of eukaryotes. Okay, remember this. What does this S mean? That is asked in the question. What does that S stands for? S stands for Swedberg unit. Either mention it as Swedberg unit if you find the option Swedberg unit. That will be the answer. If Swedberg unit is not given, then the answer would be sedimentation coefficient. Okay. Based on density, we are going to separate or see the smaller and larger subunits. Okay. Then moving forward. So this is the structure of mitochondria. The outer membrane, inner membrane. This is the space between outer membrane and inner membrane. If you observe here, this space is itself called as intermembrane space. Now it is very important. Why it is important? Because when we will be discussing the electron transport chain ETS, okay, electron transport system, that time the hydrogen ions, electrolyte ions, goes on accumulating in the intermembrane space. Okay, so it's a very important thing, important point as well. And even in examination, this is also asked the infoldings. The infoldings of the inner membrane of mitochondria is called as a cristae. Okay, remember, cristae is the infolding of inner membrane of mitochondria. Okay, and the fluid that is present in this inside the mitochondria is the matrix. Okay, now that matrix it consists of enzymes and also double-stranded circular DNA. So there is presence of its own DNA. That's why mitochondria, even chloroplast, both of these cell organelles, they are semi-autonomous. Semi-autonomous means they can replicate on their own. Okay, with the information provided by nucleus, the mitochondria and chloroplast they can replicate on their own. Okay, now these are the important things which you should remember with respect to the structure here. Then moving forward, the sectional view of chloroplast. Now, chloroplast is again having outer membrane and inner membrane. There is presence of granum, okay, thylakoid, stromal membrane. Now, what are these? We will understand. A single disc-shaped structure is called as a thylakoid. So, this is a thylakoid. Single disc-shaped structure is called as thylakoid. Now, these thylakoids are arranged one above the other and forms layers. All together, these are referred as granum. Granum means, just a second. Granum means all the thylakoids arranged one above the other is called as granum. Now, granum is singular. Granum is plural. If we are speaking about all the granums, we call it as granum. Now, these are interconnected. These thylakoids, they are interconnected with the other thylakoids. Okay. Now, this connecting part is itself called as a stroma lamellae. What is stroma lamellae here? The linking of granum with one granum with another granum is through stroma lamella. And all the fluid part that is present here, it is called as stroma. What it is called as stroma. Stroma is the fluid part that is present. This stroma consists of all the enzymes, double-stranded circular DNA as well, which are very essential for carrying out the dark reactions. Dark reactions are nothing but the process in which your food is getting synthesized. Okay, we'll come to that. We'll discuss that part in plant physiology. But remember all the light reactions which are dependent on light. If the light is present, then only the energy is produced. Those processes they are taking place in thalaphoid and stroma annuli. They are light dependent reactions and therefore they are very important in the preparation of food or in the process of photosynthesis. Then stroma, after once when the energy is produced, then that energy is utilized for the formation or next conversion of the all the inorganic or raw materials into forming a food that is glucose or sucrose. Okay, it is found in the plants. We'll discuss that process later on. So, all the enzymes that are required for that reactions are present in the stroma part. Now, remember one thing: there are three cell organelles which are having the double membrane. Okay, if a question is asked, select the following question. Select the following cell organelles which are having double membrane. So once again, I am repeating the cell organelles which are having two membranes present in them are okay. For double membrane, remember for double membrane, one is mitochondria, chloroplast. Not only these two, even chloroplast and nucleus. If in the option nucleus is not given, you can select mitochondria and chloroplast. But remember, all these three cell organelles are having two membranes. Okay, they are double membrane. If you see outer membrane, inner membrane, outer membrane, inner membrane. So all these three cell organelles they are having two membranes, double membrane structures, double membrane cell organelles. And the cell organelle which is having no membrane at all, no membrane cell organelle is a ribosome. Now that is why ribosome can be seen even in prokaryotes as well as eukaryotes. Why? Because they don't have any membrane. Okay, that's why when defining prokaryotes, we say prokaryotic cell is a cell in which the DNA is not covered by nuclear membrane and all the other membrane bound cell organelles are absent. So ribosome is not having any membrane, therefore it is non-membrane bound cell organelle can be seen both in prokaryotes and eukaryotes. Okay. So the membrane double membrane structure is mitochondria, chloroplast, and nucleus. No membrane is ribosome. Okay. And one point we'll just discuss here regarding endomembrane system. Endomembrane system, it's a coordinated function that is carried by four different cell organelles, starting from nucleus. But according to NCERT, it is the beginning of the endomembrane system. So therefore, they will not consider nucleus here. They are starting with the endoplasmic reticulum, wherein the proteins are produced. Later on, they are modified in Golgi apparatus. Then they will be forming the vacuoles and lysosomes. Okay, so these are the endomembrane system. Okay, they are very important, and that's how you should remember this. Okay, next we discussed about double membrane and monomembrane, and one more very important point about the vacuole. Okay, the membrane of vacuole is single layer. We discussed double membrane as well as no membrane. Ribosome is a cell organelle which is having no membrane at all. Mitochondria, chloroplast, and your nucleus is having double membrane. And all the cell organelles apart from these, they are having single membrane. One very important membrane name is a tonoplast. Tonoplast is the membrane of vacuoles. Okay, it's very important as the vacuole consists of all the secretions of the cell that are not required by the cell. It is all being secreted in the vacuole. So again, all the secretions will be again thrown into the vacuoles. So therefore, the vacuole membrane is very important. Okay. Then moving on to the next part, that's regarding cilia and it's for vacuole. Okay, vacuole membrane, single layer. Okay, with respect to cilia and flagella, we observe the structure. Okay, both cilia and flagella. In case of cilia and flagella, we observe the structure wherein there is central sheath having the two central microtubules. Remember, two central microtubules are there, and surrounding this central sheath there is presence of nine peripheral microtubules. How many? Nine peripheral microtubules. These nine peripheral microtubules, along with the two central tubule, this kind of arrangement is only responsible for the movement of cilia and flagella. For the movement of cilia and flagella, 9 plus 2 arrangement is very important. First. Second thing is, if you observe one peripheral microtubule, one is a complete ring structure, the other is a half ring structure. The one with the full ring structure has one structure which is coming out from it. This structure is itself called as radial spoke. What it is called as a radial spoke. Now this radial spoke, when it comes in contact with the central sheath, the complete structure starts moving. Okay, and two microtubules, they are interconnected by interdoublet bridge. Okay, that's the main thing that you should remember. One thing is about radial spoke, which structure when comes in contact with central sheath will begin the process of movement. That's regarding radial spoke. Okay, so radial spoke is a very important structure. Now, if at all, there are no central microtubules, what is going to happen? It is going to be forming the basal body, basal body of both cilia and flagella. Okay, basal body has 9 plus 0 arrangement. Because of 9 plus 0 arrangement, they cannot show any motility. The ground surface here, this is the basal body, it should not move on its own. This should be fixed. Only the upper part it should move. So this is the cilia part. Okay. Again, we call it as hook, filament. Okay. These are the basal body, filament and hook. So these are the filament and the hook is the part which should move. So these are having 9 plus 2 arrangement, whereas the basal body has 9 plus 0 arrangement, because of which there is no movement observed. Okay, this is the structure of nucleus. Now, nucleus has nuclear membrane. This is nuclear membrane. Again, nuclear membrane is made up of outer membrane as well as inner membrane. Now, presence of nuclear core is essential because the DNA and RNA should move out, it should come out from the nucleus into the cytoplasm, not only DNA but RNA. If it is carrying any message, okay, this will be seen in process of transcription. Post transcription, the RNA will be coming out into the cytoplasm for carrying out the process of translation. So translation, if it should happen, the RNA should come out from this small pore, which we call nuclear pore. The fluid that is present inside the nucleus is called as nucleoplasm. The fluid is called nucleoplasm, but here we also see the presence of thread-like structures. These thread-like structures are itself called as chromatin fibers. Okay, chromatin fiber, the thread-like structure, which is nothing but your genetic material. If it is present, it is what we call chromatin fibers. Then apart from that, we also see a very small structure, and this structure is itself called as a nucleolus. Okay, remember the protein factory is ribosomes, but the ribosomal factory is nucleolus. So RNA is being synthesized. Where RNA is synthesized? In the nucleolus. Okay, I hope it is clear. Then this is a structure of chromosome. Okay, here there are two arms. Okay, the centromere is present. Now to the centromere, there is presence of a disc-shaped structure. That disc-shaped structure is itself called as kinetochore. Okay, why kinetochore is important? Now, during the time of cell division, the spindle fibers they should come and attach to the centromere. They are not exactly going to attach to the centromere, but they are going to attach to a part of centromere, and that part is called as kinetochore. So kinetochore is that part of the centromere of the chromosome to which the spindle fibers comes and is attached. Okay, so this is very important. Remember kinetochore. Okay. Then now just to quickly remind you, the number of chromosome here is chromosome number is just one. Okay. So how many chromosomes are present here? One chromosome. How many chromosomes are present here? Okay, just draw and show. How many chromosomes are present here? Again, one only. Why? Because the chromosome number is dependent on the number of centromere, but not on the number of chromatin arms. Okay, the arms are not deciding the chromosome number, but the centromere decides the chromosome number. For example, even if I show here, okay, if I just draw you and show this structure, how many chromosomes are present here? Still one only, because there is only one centromere. Because of presence of only one centromere, the chromosome number is just one. Okay, I hope it is clear. Now, there are four types of chromosomes. How many types of chromosomes do we see? Four types of chromosomes based on position of centromere. Okay, where the centromere is placed, based on that, there are four types of chromosomes. Okay, one is metacentric. Now, metacentric is a condition in which the centromere is present exactly at the center of two chromatin arms. Now, if you see the distance here, both the distance of the arms here is the same. So, if the centromere is situated exactly at the center, it is called as metacentric. Then, if the centromere is slightly present above the central region, if you consider here, this is comparatively a smaller arm when compared to this. Okay, so slightly, if you see the center, this will be the center. It is slightly away from the center. So, such type of chromosome is called as a submetacentric. What is submetacentric chromosome? It is a chromosome in which the position of centromere is present slightly away from the center. Okay. Then here if you see, this is a very short arm and the other one is a long arm. So this is where the question gets listed. Okay. Most of the times the question asked is identify the type of chromosome if one arm is very short and the other arm is very long, that is acrocentric. Okay, and the shortest arm usually in NCERT they will mention clearly that the centromere is present exactly at this you know, terminal site, and only one of its arm is larger in length. Such type of chromosome is called as a telocentric. Okay, so remember this. Okay, metacentric exactly at the center, the centromere is present. Slightly away from the center, if the centromere is present, such type of chromosome is called as submetacentric. If the centromere is present towards the terminal side, that's ectocentric. And the chromosomes in which the centromere is present at the terminal point only, it is called as telocentric. I hope this is clear. Now, these are the some of the data given in the chapter biomolecules. Okay, so don't have to remember all of these, but you have to at least remember carbon, hydrogen, oxygen. Okay, these are very important three elements. So the amount of hydrogen present in Earth crust is 0.14, and in our body 0.5. Carbon in Earth crust only 0.03%, but in our body 18.5. Amount of oxygen that we see in Earth crust is 46.6. In our body it is 65.0. Now remember, the most abundant element in the Earth crust is oxygen. Okay, next to that is silicon. Okay, and very important point regarding this. And then if we come for some of the inorganic constituents, sodium, potassium, calcium, magnesium. These are some of the important inorganic constituents present in our living tissue. Okay, for the transfer of the signals, non-impulses, we require sodium voltage gating channel, potassium voltage gating channel, and calcium. As you know, calcium are very essential for the binding to serotonin. Okay, we discussed that in locomotion, and calcium is a very important role. Okay, magnesium, as you all know, for the process of respiration. During the process of respiration, there is a requirement of magnesium in the link reaction. We discussed about that. Okay, so again, magnesium is very essential. Water, obviously, majority or most of our cells, the composition of our cell is nothing but water. Okay, that's a very important point. Now, here the general structure of amino acid is C R C double O H NH2 H. Now, here the general formula of amino acid. Why it was called as amino acid? Because it consists of the amine group on one side and acid because it is having carboxylic acid on another group. Because of which the name was given as amino acid. And this is the general form of amino acid. Now, the amino acid kind of acts like an acid in a basic solution and acts as a base in case of a acid solution. Why? Because it has the ability to donate a proton as well as to accept a proton. Speaking from chemistry point of view, because now we are learning biochemistry here, that's biomolecules. So you must remember that here a proton is getting accepted. In condition of proton is getting accepted, because of which NH2 became NH3 plus. An extra additional proton is added to a amine group. Now, Obviously, if it is accepting a proton, then this will be nothing but this will be acting as a base. Okay, basic. Then in the C condition, here the proton was donated. Okay, the proton was donated here. Okay, the compound which donates a proton is nothing but a acid, according to Lonsted Brody concept of the acid base. We say that the proton which is being given, the proton donors are called as acids and proton acceptors are called as base. So this condition represents the Zwitter ion form. Zwitter ion form is nothing but it's a representation wherein the partial, the proton is being given and the proton is being accepted. Okay, so that form, okay, of amino acids which can donate a proton and accept a proton is itself referred as Zwitter ion. That this central condition is itself called as Zwitter ion. Okay. Now these are some of the structures given in your NCRT. Make sure you remember it's a hexose sugar. Glucose is an hexose sugar, whereas ribose is a pentose sugar. Hexose sugar, wherein oxygen is present. The first carbon here, second carbon, third, fourth, fifth, and sixth carbon is present here. Okay, remember this. Okay. In case of pentose sugar, this is very important structure because we also discussed the same structure in case of molecular basis of inheritance in the second year as well. Okay, so here, if at all the oxygen is present on the second carbon, we say it as the ribose sugar. If oxygen is absent on the second carbon of the pentose sugar, we call it as the deoxyribose sugar. That's a sugar present in DNA. If at all oxygen is present, it is called as ribose sugar present in RNA. Okay. Now, not only that carbohydrates, but we'll also discuss with respect to some other points as well. We also discuss some basic amino acid structures such as glycine, alanine, as well as serine, which is an alcoholic amino acid. We learn the structure as well. The basic amino acid is glycine, which is present in the form of C, the same, this as discussed here. Now, here this R here in case of glycine is H, and it's a primitive amino acid, the first basic amino acid, and this amino acid is symmetric or asymmetric. It's a asymmetric compound. It's called what we call it as optically inactive. The only amino acid which is optically inactive because of absence of four different groups on the same carbon atom. It is asymmetric. Okay, so because of which this glycine is optically inactive. Only amino acid to be inactive is glycine. Then alanine, just replace this H with CH3. Now, when you replace the H with CH3, it becomes alanine, and this is optically active because the all the four compounds that are attached to the carbon are different. Therefore, this compound is optically active. Okay, and if you just change this CH3 to CH2 OH, this becomes serine. Okay, alcoholic amino acids. Okay, so again there is a list of amino acids. 20 different amino acids are present. Among 20, the structures that you should remember in the biology is three: glycine, alanine, and serine. Okay, just very simple. If you have to remember again, and just write it down here. The general form of amino acid is COOH, NH2, H. Now, in place of R, if we write H, this becomes glycine. Okay. Now, the same thing, everything is the same. Okay. C, COOH, NH2, H. Now, here in place of R, if we write CH3, okay, if you write CH3, it is nothing but it is alanine. Okay, the same thing here. Again, one more time. Right, everything the same. COOH, NH2. Now here you write H, and here instead of writing CH3, you write CH2OH. This becomes serine. Okay, I hope it is clear. Next, this is the structure of a 16 carbon fatty acid, okay, which we call it as palmitic acid. Palmitic acid is a 16 carbon compound here. This is first, and this is 14, and this is 16. A total of 16 carbon compound fatty acid, that's palmitic acid, is present. Glycerol is present. Now, when fatty acids and glycerol combine, it forms lipid. Okay, if only one fatty acid combines here, it is called as monoglyceraldehyde. Okay, then monoglyceride for that matter. If two fatty acids, similarly, if one more fatty acid is present here, if that combines with the OH group, again it is called as a diglyceride. If three groups combine, it is called as a triglyceride. Okay, so it is as simple as it is. Okay, if this is glycerol only, the water molecule gets eliminated here. Okay, OH from this side and H from this side will get eliminated, and as you can see, the O remains as it is, C double bond O remains as it is, and rest all is R. Okay, so that's how we say it as a triglyceride. Triglyceride means glycerol plus fatty acids. Glycerols plus fatty acids will form the lipids. Okay, simple lipids. Then there is a phospholipid which we call lecithin. Phospholipid, remember, it's a very important component of plasma membrane. So plasma membrane is made up of lipids only, bilipid layer. The major component there is phospholipid, and the example for phospholipid here is lecithin. Okay, remember that. Okay, don't need to remember the structure here. Just you have to remember the name lecithin. Okay. Then, with respect to cholesterol, remember two honeycomb mazes, one and two completely. These are two honeycomb mazes. Again, there is two more honeycomb mazes. So if you remember like honeycomb, then you can remember the cholesterol structure. Simple trick to remember the structure of cholesterol. Then adenine, purines. Okay, this has been asked in your NEET. Finally, also select the purine. Okay, you must be very cautious, very careful in selecting the name of purine, selecting the name of a nucleoside, and selecting the name of a nucleotide. They, they are all differing very slightly, but the name confuses you. Okay, now remember adenine. Okay, it's a purine. Who are pure? AG or G is pure. You have to remember like that. Purines. Who are pure? A and G. They both are pure. Adenine and binding, they are purines. Then who will come under pyrimidines? In case of DNA, it is T and C. In case of RNA, it is U and C. Okay. The C is a question if it comes, which is the pyrimidine that is found both in DNA and RNA. The answer will be cytosine. Remember that. And which is the only pyrimidine which is present in RNA? That's uracil. Okay, uracil. Okay, name is very important again. Now, to this uracil, if the compound uracil gets added to the pentose sugar, here in case they have taken the ribose sugar because on the second carbon there is presence of oxygen. If oxygen is present, we have to identify it as ribose sugar. So, the sugar that is present here is ribose sugar, irrespective of whether it is ribose sugar or deoxyribose sugar. If the uracil gets attached to the pentose sugar, it is called as uridine. Now, the name changes. Select the nucleoside among the following. They will give you uracil, uridine, and uridinate acid. The three will be slightly confusing. You should not get confused. Uracil is the name of the pyrimidine. Uridine is the name of the nucleoside. Side. What is actually nucleoside? Sugar plus nitrogenous base. What is nucleoside? Sugar plus nitrogenous base. Sugar plus nitrogenous base. Which is our nitrogenous base here? Uracil. So uracil gets attached to the first carbon of the pentose sugar. Then this structure is itself called as nucleoside. Now, what is nucleotide then? Sugar, nitrogenous base. Anyhow, sugar will be present and nitrogenous base will also be present. For that sugar, for the fifth carbon, on the fifth carbon, if we add a phosphate group, okay, if we add a phosphate group, it is itself called as a nucleotide. So, what is nucleotide? Sugar plus nitrogenous base plus phosphate group. All three, when they are present, it is itself called as a nucleotide. Adenine is a purine. Adenosine is a nucleoside. Adenylic acid is a nucleotide. It's a very important concept, confusing concept, but very easy. Try to remember adenine, nin, sin, ilic. Okay, nin. First is yours, nin, yours. Then sign will come. Okay, your sign. Then for ilic acid. Okay, so you have to remember your sign acid. Okay, that's nin, adenine, guanine. If you have to name it for guanine, it's guanine, the nitrogenous base name. Guanosine is the name of the nucleoside. Guanylate acid is the name of the nucleotide. The same goes with pyridates as well. Next, this is very important table. Very very important table. Why? Because first thing is the secondary metabolites. Okay, you have to remember the name. Okay, pigment means the color producing secondary metabolites. What are actually secondary metabolites? If we have to discuss in short, metabolites are itself the products, intermediate products formed during the process of metabolism. So either during the construction process or during the destruction process, either in the making of molecules or in the breaking of molecules. That is anabolism and catabolism. There are some products which are going to be produced in the middle. Okay, those products are itself called as metabolites. Then what are primary metabolites? Directly going for secondary metabolites. Primary metabolites means they are the metabolites which are naturally utilized by the cell. For example, amino acids, okay, glucose molecules. These are all the compounds which are naturally utilized. They are naturally utilized or they are naturally present in the or they are directly involved in the growth and development of the organism. Primary metabolites, all the monosaccharides, okay, all the nucleotides, okay, all the simple amino acids, okay, all the monomers which are very essential, all the micromolecules, microbial molecules, they are all coming under primary metabolites. And the secondary metabolites are the metabolites which are again produced during the course of metabolism. Those metabolites, okay, they can be either pigments, alkaloids, terpenoids, essential oil, toxins, lecithins, drugs, polymeric substance. Okay, pigment, carotenoids. I hope you can remember carotenoids in the next chapters. That's in the plant physiology. We will be discussing about carotenoid as a compound. Okay, pigment. You can remember that. Anthocyanin is a one more very important pigment present usually present in the beetroots and all. The color for the beetroot is mainly because of anthocyanin pigment. So they are all pigments. Alkaloids, morphine, codeine. I hope you people remember. These are all alkaloids, drugs. Okay. The terpenoids, monoterpenes, diterpenes. The name is itself suggesting. You don't have to remember this. Terpenes, terpene, monoamine. They're all ending with the same name. Terpenes, terpenoids. They're all coming under terpenoids. Oils. The name is itself suggesting lemongrass oil. So you don't have to remember it further. The essential oil among the following: lemongrass oil, nami, the name is itself suggesting. Toxins, okay, albin and dressing, in, tox in. That's how you can remember. Okay, tox in is there here also. The name ends with resin, albin, toxin. You can just cancel out this S, toxins. Remember toxin, toxin, iron ends with in, here also ends with in. That's how you can remember. Lecithin, okay. Lecithin, again, chlorquinoline, A, again, very important. Drugs, vinblastin, anti-cancerous, curcumin, okay. These are two drugs that we obtain from the plants. Polymeric substances, maybe rubber, gums, cellulose. These are all the polymers. Okay. The components, the average composition of cells, the major component is water around 70 to 80%, proteins around 10 to 15%. These are the two things which you should remember. And apart from that, nucleic acid 5 to 7%. Okay. These are the data based question which you should remember as this. Then moving forward, also this table is again very important. Several times the question has appeared directly. What is the role of collagen? It acts as intercellular ground substance. So to hold the cells, okay, collagen plays a very important role. Okay, and it's the most abundant protein in the animal world is collagen. Okay, have to remember. Most abundant protein in the plant world is rubisco. Okay, most abundant carbohydrate in the plant world. Okay, it is your cellulose. The most abundant carbohydrate in case of animal world is glycogen. Okay, remember that. Then trypsin is an enzyme. Obviously, you people have studied in 10th standard as well. Trypsin, where it plays a very important role in the process of digestion. Insulin. Here there are two things. One is insulin, the other is inulin. Don't get confused between insulin and inulin. Okay, insulin is a hormone. Inulin is a carbohydrate. Okay, it's a homopolymer of fructose, alpha D fructose. Then antibodies. These are all proteins. Remember, okay, collagen, trypsin, insulin, antibodies, receptors, VF4, these are all proteins itself. We are seeing their function. Antibody, it finds against the antigens. Okay, receptors, they are responsible for carrying out any reactions taking place. If the receptors are absent, then the cells will not get any signals. Okay, if there is no signal, then there is no further transmission of information. So receptors are very essential. Those receptors can be formed from the proteins. And GLUT4, that's glucose transport number 4 molecule, that is essential in transporting the glucose into the cell. Okay, so glucose, if it has to enter into cell, it requires one protein which we call GLUT4, GLUT4. Very important. Okay, this is essential for transport of glucose into the cells, and also remember the collagen. Okay, I hope it is clear. Okay, now is the image not clear? Is the image not clear? Just check and respond once. The audio is not clear or the video is not clear? Just check and respond once. The audio is not clear or the video is not clear? Okay, I resume that. It is clear and I'll go forward. So this is the diagrammatic representation of a portion of glycogen. So this is complete portion of glycogen. Okay. So whatever the branches you are able to see, these are all the structure of glycogen. Okay. Glycogen is the reserve food material in case of animals. So glycogen is the reserve food material in case of animals. Okay. Now glycogen along with the starch, glycogen along with starch, they show a positive test for iodine. Now remember, speaking from chemistry point of view, all the monosaccharides, they give positive test for Fehling's. Okay. If you go for Fehling's test to find that whether the sugar is reducing or non-reducing, you will find that all the monosaccharides, okay, which have either one molecule of aldehydes or your ketone, they will obviously produce what, the reducing sugar. All the monosaccharides they will be reducing, and therefore they give positive Fehling's test. If at all, if you consider the disaccharides, okay, disaccharides, your lactose and your one more, one is lactose sugar, the other is sucrose, okay. The other one being, one is maltose, right? Another one is maltose. Lactose and maltose will also give positive Fehling's test, but sucrose will give negative Fehling's test. Okay, sucrose, which is also called as invert sugar, okay, is not having any reducing sugar, therefore it gives negative Fehling's test. Now, for glycogen and starch, we cannot use the same sucrose test. I mean the same Fehling's test. So for glycogen and starch, we use iodine test. We prefer iodine test. Now, glycogen and starch both are having helical structure. Okay, both are having helical structure. Because of presence of helical structure, the secondary helical structure, these two molecules can hold the iodine. If they can hold the iodine, then it gives a positive iodine test. But remember here only, cellulose will not show the test of iodine because it is not helical. As it is not helical, so it cannot hold iodine molecules. So iodine test is not given. Okay, just a sec. So, as we are discussing, the glycogen and starch, they both are having helical structure because of which there is positive iodine test that they show, and cellulose that is not having helical structure because of which the cellulose will not show iodine test. Okay, I hope this is clear. Now, the structures of proteins. There is a primary structure, secondary structure, tertiary, and quaternary structure. Now, these are the four different structures that we observe here. Primary structure, in primary structures, all the amino acids are arranged in a linear chain. If the amino acids are arranged in a linear chain structure, then such a structure is said to be primary. Okay? There is no poly and there is no involvement of other bonds other than the peptide bond. So in between two amino acids there is presence of peptide bond. Remember this point. Between two amino acids there is presence of peptide bond. The only bond that we can see here in case of primary structure. Now, the secondary structure, it can be alpha helix or beta plated sheets. Alpha helix, okay, it's a helical structure. And it's always right-handed helix, you should remember. Now, in this alpha structure, we can also see the intra-hydrogen bonding, not inter. You have to understand the difference between inter and intra. Intra means within, and inter means in between. So here in alpha helix structure we see intra hydrogen bonds, and beta plated sheets we see inter hydrogen bonding. So apart from peptide bond we also see hydrogen bond in the secondary structures. Now tertiary structure, it's a very important biological structure because most of the enzymes, biologically active processes, they all will be taking place in which structure? That is tertiary structure. Okay, for carrying out all the important biological processes, the structure that is very essential is tertiary structure. It has the presence of even the other bonds as well. The disulfide bonds, inter-hydrogen bonds, inter-hydrogen bonds, not just disulfide bridges, but even the other bonds such as van der Waals was potential, even iron bond can take place in the tertiary structure. They also have presence of active sites. Active sites, as you know, if you have studied the enzymes, active sites is a site to which the substrate will come and bind. So, if the enzymes, if should they carry out, they should carry out their function, they should have that active site, and those active sites are usually present in the tertiary structure. Then quaternary structure. Okay, quaternary structure we usually see it with respect to heme group. Okay, the hemoglobin contains four heme group. Okay, that heme group which is present, which is acting as a central compound. Okay, in all the globin chains. Okay, two alpha and two beta globin chains together, and the center there is presence of heme group. So in the quaternary structure, it is an example for heme group. Okay, the example here is heme group. Remember that. Okay, then this is very important. The enzyme, they have discussed about one enzyme which is called as carbonic acid, carbonic anhydrase. What is this carbonic anhydrase enzyme? Why is it that essential? In the conversion of carbohydrate, when carbohydrate combines with water in presence of carbonic acid, it will form a carbonic acid. And further, this is not the end product. Further, carbonic acid again in presence of carbonic anhydrase, it is going to form H plus HCO3 minus. Now, it is going to form the bicarbonate. Okay, H plus plus HCO3 minus. Now, that's a structure or that's a ion which is going to be formed for transport of carbon dioxide. Now, if you remember or recall the transportation of carbon dioxide, the transportation of carbon dioxide can occur in three forms. One is carb amino hemoglobin. Okay, that's the 23% of carb amino hemoglobin is transported via the hemoglobin, and nearly 70% of the carbon dioxide is transported via the using of the carbonic bicarbonate ions, as they are forming the ion form. They are easily soluble in the plasma and it is directly transported to the lungs. And remaining 7% it is dissolved in the plasma, and in the plasma itself the carbon dioxide is taken to the lungs. So nearly 77% of the carbon dioxide will be carried in the plasma itself. Okay. So for doing that, carbonic anhydrase, this enzyme is very essential. Now, NCERT has given some data which says that carbonic acid, if this enzyme is absent, no doubt the carbonic acid is formed, but only 200 molecules per hour when in absence. In absence, 200 molecules are going to be produced per hour. But if at all this enzyme is present, it is going to form nearly 600 molecules per second. So where is lakh and where is just 200 digit? And whatever we are talking, the number here 200, is it with respect to hour, 1 hour? And in presence of this enzyme, it is going to produce lakhs of carbonic acid molecules per second. So that's why enzymes play a very important role in our biological processes. Okay. Now this is a common glycolysis process wherein one molecule of glucose is converted into two molecules of pyruvic acid, which we'll be discussing here. They have given the formula for pyruvic acid, which you should remember here. Okay. Pyruvic acid is nothing but C3H4O3. Okay, this is the molecular structure for the structural formula for pyruvic acid is C3H4O3. You should remember this. Okay. Then this is again why we have mentioned here in biomolecules is because this is a 10-step process. Okay, 10-step process, and all the processes they require enzymes for the conversion of glucose to glucose 6-phosphate will be requiring heterokinase. Okay, if the enzyme is not present, then obviously the process is not going to go forward. So all the enzymes are very essential for the process to take place. Okay. Then this is concept of activation energy. Now, what is activation energy here? The substrate, okay, are converted into product. So this is a natural process wherein the substrate should get converted into products. If at all enzymes are present, okay, if at all enzymes are present, this substrate can be converted into product at a very low energy. Now, for any process to begin, there should be requirement of some energy. Okay. Now you are pursuing NEET. So to crack NEET, you need a little motivation. You need little fire within you. So somebody has to light that fire. Right? So this activation energy acts like that fire here. Okay. So activation energy is needed. It's a minimum energy that is required for the conversion of substrate into product. If enzyme is there, the energy that is required for converting substrate into product is very less. And if at all the enzyme is absent, the requirement of energy for converting substrate into product is very high. So therefore, the enzyme is very essential. Okay, that's one thing. And remember that this is exothermic process. Okay. If at all a question comes by observing this graph, either point out A or B. This is it. You have to either point with respect to A and B, identifying A and B. So they might give you options such as A is with enzyme, B is without enzyme, with enzyme but exothermic, with enzyme endothermic. Okay. Again A plus endo, B plus exo. So the correct option that you should choose when they ask with respect to enzyme. If enzyme is present, then the correct answer would be now this. Yes, A, this one. Okay. So this is for activation energy that is required for by the enzyme for converting substrate into product, and this is a exothermic reaction. I hope this is clear till here. Then moving forward. Now this is with respect to factors affecting the uh, concentration or factors that are responsible for the enzyme activity. Okay, that affects the normal functioning of the enzyme. pH, temperature, and substrate concentration of substrate. pH, if it is very less, the enzyme activity is again very less. If at all, if it is the pH is very more, the enzyme activity of the enzyme is again reduced. If the pH is optimum, then the enzyme efficiency is maximum. So enzyme efficiency is maximum at optimum temperature. Similarly, the enzyme efficiency is maximum at again optimum temperature. If at all there is less temperature, one point that you should remember here, less temperature leads to inactivation of the enzyme. Okay, high temperature will degrade the enzyme. Remember this. Okay. Then also the last is substrate concentration. S stands for concentration of substrate. If there is more substrate, then what happens? The velocity of reaction, that is rate of reaction, goes on increasing. Okay. As there is increase in substrate, the velocity of reaction also goes on increasing only up to certain point. Okay, only up to certain point. There is something called as Vmax. If at all the Vmax, the maximum efficiency of the enzyme is reached, even after reaching the maximum efficiency of the enzyme, if you increase the substrate concentration also, the efficiency will not go beyond this line. Okay, the efficiency of the enzyme will be constant at that time. So half V max by 2 is where we see the half potential or half efficiency of the enzyme at a very low substrate concentration, and if you require maximum concentration, V max is the substrate concentration should be more. Okay, these are some factors which affects the activity of the enzyme. Okay, this is again very important. That is nomenclature of enzymes, say nomenclature of enzymes, say types of enzymes. Okay, so there are 6 types of enzymes mentioned in your NCERT. That's oxidoreductase, which is also called as a dehydrogenase. It's an enzyme which brings about oxidation and the reduction. So consider one substrate, S and S dash as a two substrate. If S is reduced, by adding the enzyme, I am making S oxidized. So substrate is going to get oxidized in presence of oxidoreductase. S dash is going to get reduced. Okay, so we are just oxidizing and reducing. Okay, so simultaneously, if we use this enzyme, that is what we are going to say it as the oxidoreductase enzyme. Okay, then transferase, as the name is itself suggesting transfer. Okay, nothing but a group is getting the transfer. That's it. Okay, the functional group that is present or that is found attached to the substrate here, S dash is going to join or combine with S and G is going to come out. That is what we call it as a transferase. That function, that work is carried out by transferase enzyme. Other than hydrogen, that's very important. If at all that G, that group is a hydrogen which you want to remove, it is not possible with the help of transferase. But if at all there is other compound or functional group which you want to remove from that particular group, you can use the transferase enzyme. Hydrolases, the name is itself suggesting. So here the hydrolysis, okay, removal of water molecule. Okay, by removal of water molecule, there was a question here. Remember, if you want to form the sucrose molecule, okay, sucrose. What is the formula for sucrose? Okay, glucose plus fructose. Okay, glucose C6H12O6 plus fructose again C6H12O6. The total should have actually it should have been C12H22O12. Okay, sorry, OH24O12. So this should have been the formula, but this will not be our formula. Okay, C12 H22O11 plus H2O. So a molecule of water is getting eliminated here. That elimination of water molecule by combining the two molecules is what we call hydrolysis. That is carried out by hydrolysis enzyme. Lysis is an enzyme that will help break the groups that is present within a compound, and they are going to help in the formation of bond between those compounds, and ultimately results a carbon-carbon double bond here. It leaves carbon-carbon double bond, and there is also formation of XY compound. Okay, so that is done by, okay, that work is done by lysis. Okay, so lysis carries out that function. Then isomerases, the name is itself suggesting isomerases means it is going to form the different types of isomers. Okay, it may be geometric isomers, positional isomers, the optical isomers. Say for example, if you get trans group, any trans compound, if you want to change it into cis form, we are going to use which form, which enzyme? Isomerases. Okay, so the interconversion of cis and trans forms is also possible by the use of isomerases. I hope you are familiar with all the cis and trans forms that comes in optical isomerism. Okay, then sorry, geometric isomerism as well. Then there are even positional isomers. All these different isomers are present, which helps in interconversion of compounds. Okay, ligases. Okay, ligase is, as you all know, DNA ligase itself is joining, right? So this enzyme is very essential for joining of or catalyzing of different compounds, which links together two compounds. Okay, for linking of two compounds, DNA ligase is very essential. That's regarding the enzyme part. That's biomolecules part. Okay. Now we'll move on to the 10th chapter. That's a cell cycle inside division. Okay. This is very important. G1 phase, S phase, G2 phase, and prophase, metaphase, anaphase, telophase, cytokinesis. The first four, they are called as karyokinesis, last is cytokinesis, and here we will see the G0 phase. They enter into inactive state. G1, S, and G2. All these three are referred as interphase, or also called as resting phase, during which the cell will prepare itself for undergoing M phase. Okay. G1 is the phase in which the RNA, the protein synthesis, the cell starts to grow. S phase is that particular phase in which the amount of DNA gets doubled. Very important. Amount of DNA gets doubled. The amount of DNA gets doubled, but the chromosome number remains the same. That's the catch here. Okay, if the chromosome number is 46, it remains 46, but the amount of DNA gets doubled. So the DNA replication takes place in the S phase of interphase. Okay, not only this, in case of animal cell, there is a duplication of centrioles as well. That also takes place in the S stage. So in G2 phase, again the same thing. That is, the cell size goes on increasing. The RNA and the proteins, they are synthesized. Okay, and finally they get ready to enter into the prophase. Okay. Now the M phase, when we begin of M phase, before that, just remember the time duration required for the completion of cell cycle. In case of yeast is 90 minutes, and in case of human beings it is 24 hours. Okay, wherein 95% of the time the cell cycle will be present in the interface. Hardly 5% of our cell cycle is for young phase, wherein the actual division takes place. Those are some important points which you should remember. Then, when we start off the mitosis there is early prophase and late prophase. What is the simple point that you should remember here? The nuclear membrane, nucleolus disappears. Okay, and slowly the chromosomes they start to condense. Chromatin fibers start to condense to form chromosomes. So these are chromatin fibers, thread-like structures. Slowly, at the late prophase, we find that the chromosomes are being formed. Okay. Now here there is no what exchange of genetic material. There is directly the chromosomes are formed here in the late prophase. Then what happens? Transition to metaphase. Slowly the centrioles will also start moving towards the opposite poles. Okay. And during the metaphase, in the early metaphase stage, the centrioles will produce the asters, and these asters are responsible for the production of spindle fibers. Now these spindle fibers goes and attaches to the kinetochore of the centromere. We have discussed that where they exactly attached, not to centromere, exactly centromere, but centromere has one more part which we call it as the kinetochore. So spindle fibers they get directly attached to the kinetochore of the centromere, and all the chromosomes they align in one single plane, and that single plane is itself called as a metaphase plane, or we also call it as equatorial plane, wherein we see that all the chromosomes they align in a one single plane. And metaphase is the best phase. Metaphase is the best phase to study the structure, behavior of the chromosomes. Okay, that's the only phase wherein we can see all the chromosome at its best. Okay, the structure is clearly visible. Then those metaphase then further starts condensing. Okay, the spindle fiber starts condensing. As a result, the spindle fibers, when they start condensing, the centromere will split in the anaphase. So in the anaphase, what happens? The centromere will split, because of which now we can see there are total of 8 centromeres here. Okay, 4 on one end, 4 on another end, right? Earlier we saw that here there was how many chromosomes? 1, 2, 3 and 4. 4 chromosomes were there. But after the end of anaphase or during the anaphase, because of splitting of centromere, the chromosome number increases. Now earlier it was 4, now it became 8. But 8 how it is present? 4 on one side, 4 on another side. And that is how the chromosome number is maintained. Okay, if you see mitosis is called a repeational division for a reason. Why? Because it maintains the chromosome number. Okay, so again in anaphase, all the chromosomes will move towards the opposite pole, and in the telophase, slowly what happens? The chromosomes they start to decondense and form the fibers, thread-like structure. Chromatin fibers are formed. Again, there is reappearance of nuclear membrane, nucleolus, and slowly the centrioles start to degenerate, and all the cell RNA they start to reappear. Now, here we should remember about two points. Okay, one is the cytokinesis. Okay, interphase is with respect to your, okay, two stages between two M phase there is interphase. Okay, remember that. But the cytokinesis in a plant cell occurs by formation of cell plate, whereas cytokinesis in case of animals it occurs by formation of a cell furrow. Okay, that is what you should remember. Cell furrow usually what happens from the externally the cell will divide into two. Okay, from externally there is a force because of which the cells will divide into two. Whereas in case of plant cell, you know that the cell wall is very rigid, it is very hard. And in animal cell, there is no requirement of cell wall. The atmosphere is cell membrane. So in case of plants, because of presence of cell wall, that cell is very hard from outer side to inner side. It cannot divide. So what happens in plant cell? The cell plate will start forming. In case of plant cell, there is formation of cell plate in the center. So from the center, it goes to the peripheral side and creates a two division. Okay, that's what you should remember. And interphase is a phase between two successive M phases. Should remember that between two successive M phases, there is interphase. Now, with respect to meiosis 1, there is prophase 1, metaphase 1, anaphase 1, and telophase 1. It's absolutely very simple, but it's not that simple. Prophase 1 is again having 5 substages: leptotene, zygotene, pachytene, diplotene, dikinesis. Leptotene is a stage in which the chromosomes they start to condense, and the chromatin fibers start to condense and form chromosomes. So chromosomes are formed. What happens in zygotene? Very important three features. Three things happens in zygotene. One, pairing of homologous chromosome. If you want me to write, I will just. Yes. So in case of leptotene, there is condensation. Leptotene then followed by zygotene. In zygotene, first point is pairing of homologous chromosome. Now, not just pairing of homologous chromosome, that pairing of homologous chromosome is itself called as synapses. And synapses, because of formation of the two chromosomes together, it is also going to form synaptonymal complex. Because of formation of synaptonymal complex, the homologous chromosome will stick together. Okay, that is what is going to happen in zygotene. Then pachytene. Now, pachytene, the name is itself suggesting that there is a patching that is taking place. Pachytene, that means exchange of genetic material between paternal and maternal chromosome. Okay. Now the point of crossing over is called as chiasm, but we'll not discuss about that. Here there is complete formation of bivalent structure. Bivalent and tetrad is going to be formed, but I hope that you will remember. Then here one more very important point that you should remember here is this process is itself called as recombination, and it is going to take place in presence of an enzyme called as recombinase. So it's an enzyme which is involved in the process of recombination. Now, after patchotin, there is the next stage is diplotin. Now, diplotin is that stage wherein the synaptonymal complex gets dissolved. So that is what we call dissolution of synaptonymal complex. Okay. Now, because of dissolution of synaptonymal complex, the homologous chromosomes they start to get separated, except the point of crossing over. That point of crossing over is called as chiasma. Okay, chiasmata. Okay, point of crossing over. That point is still present. Okay, means that attachment is still present. Now, the last stage is diakinesis. What happens in diakinesis? Terminalization, terminalization of chiasma. That is what is going to happen in diakinesis. Okay, so these are the five substages, and they are the key points of five substages that takes place in prophase first of meiosis. But also remember some of the oocytes. If you remember primary oocytes, in case of mammals, they get arrested in prophase 1. Specifically, they are going to get arrested in the diplotin of prophase 1. Now, this is prophase 2, metaphase 2, anaphase 2, telophase 2. One point that you should again remember here is, in case of anaphase 1, here there is no splitting of centromere. So, anaphase 1 will not show any splitting of centromere. Remember this. And that is why the chromosome number gets reduced. If you see earlier in a single cell there were a total of 4 chromosomes, but at the end of telophase 1 in a single cell we only see 2 chromosomes. Why? Because in anaphase the chromosomes were not divided. It is in anaphase 1 that the centromere did not split. As the centromere did not split, the chromosome number was reduced to half. Okay, so in case of anaphase 2, it is similar to that of mitosis. Okay, wherein the centromere will split. If it splits, again it is going to form the equal number of chromosomes. Already the number was reduced to half here, so the haploid number goes on continuing, and ultimately in all the 4 cells we see haploid number of chromosomes. Okay, so as there is haploid number of chromosomes, you must know that at the end of meiosis we get 4 haploid cells. Okay, 4 haploid cells are present. Usually the meiosis process occurs in meiocytes. What are meiocytes? The gamete producing cells are called as meiocytes. Okay, next. Now, that completes your the third unit. We will move on to the fourth unit, that is plant physiology. Okay, plant physiology. Here, initially you see all the different experiments, right? Now, this is first Priestley Joseph experiment, wherein he found out that in a bell jar, this is also called as bell jar experiment, wherein he keeps mice and a burning candle. Okay, both are producing carbon dioxide. For the candle to burn, it requires oxygen, and for the mice to stay alive, it also requires oxygen. But where the oxygen source is coming from? Nowhere. Okay, so in some time, what happens? The mice is going to die, and also the candle is going to turn off. Why? because there is lack of oxygen. For burning also we require oxygen. For the mice to breathe also we require oxygen. Then when precisely he keeps a plant in the bell jar, what happens? It is going to produce oxygen, and oxygen is utilized by the candle for burning, and also it is utilized by the mice for respiration. Now, from the candle, okay, what is going to happen? The candle is going to give carbon dioxide to the plants. Not just the candle, but even the mice is also going to give carbon dioxide to the plant. So plant is going to utilize carbon dioxide in preparation of the food to carry out the process of photosynthesis. So all these are three, that's plant, candle as well as the mice. All three will be and will be carrying out the regular activities. But once when the candle burning stops, again it has to be replaced so that the process again continues. Next. So this is a structure of chloroplast that we are seeing here. The chloroplast is again having outer membrane and inner membrane. The presence of grana, okay, grana is present, and as we discussed, between two grana there is presence of stroma lamellae, okay, that is connecting two granas. There is also presence of ribosomes. There is presence of starch granules. There is presence of lipid droplet. Not only that, there is also presence of circular DNA as well. Okay, there is presence of circular DNA and all the different enzymes. All they are present here. Okay, and stroma is responsible for carrying out the dark reactions. So we'll discuss it one by one as we discussed earlier. Then this is the graph, okay, that explains about how the pigments they are showing the process of photosynthesis at which wavelength. Okay, so usually it was given in one single line, but we'll write here 400 followed by 500 here, then 600. I guess the marking is done. Yes. So 400, 500, 600, 700. Now, if you see, observe very carefully, these are the pigments, different pigments that are showing photosynthesis at different wavelength. All the pigments cannot absorb the same wavelength of light, right? So here there are four different groups that we are observing, and all the four different groups of pigments they show different lines. Here they have discussed only about three pigments. Okay, they have left out xanthophyll. Now, if you remember, chlorophyll A is a blue line. Okay, so the blue line, whatever you are observing, it is for chlorophyll A. Now, chlorophyll A, the graph or the peak that we obtain is at two different wavelengths. So, one is at 420, somewhere around 420 and half. The other is, just a second. Okay. So one is around 420, the other is around 680 nanometer. So we'll see that if you see here, and the other line is around 680 nanometer. Okay. If we look at chlorophyll B, the first peak that we are observing, major peak that we are observing is somewhere around 480. And the second small peak, it's a very small peak when compared to chlorophyll A. It is also observed somewhere around 640 nanometer. Okay. Then the carotenoids, they are showing the maximum peak in the range of 460 to 480 nanometer. And we observed even the normal photosynthetic process, where actually the maximum, at which wavelength the photosynthesis is maximum. So we obtained again two peaks, around one around 420 and the other around 680 nanometers. And we combined the first data with the second data. We overlapped these data and we got a third graph. Okay, this is the third graph. So the pigment which shows the similar readings, the among the three pigments which showed the similar readings was actually found out to be chlorophyll A. So chlorophyll A was carrying out the process of photosynthesis and the same or more or less the same wavelength. So therefore, we came to the conclusion that the reaction center, the actual molecule which is involved in carrying out the process of photosynthesis, the major pigment is chlorophyll A. So that is how we came to the conclusion. So this is what they are explaining here. The light harvesting complex. This is a complete complex molecule here. There are pigment molecules. Okay. This is a reaction center. Reaction center wherein the major pigment is present, chlorophyll A. These are pigment molecules along with proteins. Okay. Pigment molecules may be carotenoids, chlorophyll B, chlorophyll C. Okay. Xanthophylls, chlorophyll D. All these pigments are present. Okay. They are acting as a pigment molecule. The major pigment is chlorophyll A, which is called as reaction center. Okay. Now all together it is referred as light harvesting complex. So LHC refers to light harvesting complex, which includes pigment molecules and the reaction center. Okay. Now set scheme of light reaction, which is also called as non-cyclic photophosphorylation. It's a process in which the photons are absorbed by the light harvesting complex, and all the pigment molecules they carry the light and they transfer the electron to the adjacent molecules, and ultimately the energy is given to the reaction center molecule, wherein there is a photolysis of water, very important. Now don't follow this one, this data. Actually we cannot say one nascent oxygen is produced. So the two molecules of water, it undergoes photolysis. Photo means not the photo that you take, the photo means sunlight. Lysis means breakdown. So, in presence of sunlight, the water molecule is broken down into 4 protons, 4 electrons, and 1 molecule of oxygen. Okay, so that is very important, and this is the oxygen that has been utilized in the respiration process, which we discussed later on. Okay, now this water molecule, what happens here is this water molecule will be broken down to produce protons and electrons. The electrons are utilized, not the protons. The proton role is very important, which I will tell you. The electrons are utilized and they are sent to electron acceptor, primary electron acceptor. Okay, so paradoxin reducing substance followed by the cytochrome complexes. They are all the acceptors which we speak here. First will be photosystem 2. Remember, that's very important. For photosystem 2. Photosystem 2 occurs at 680. Photosystem 1 occurs at 700 nanometer. Okay, remember this. Okay, photosystem 1 is taking place at 700 nanometer and photosystem 2 at 680 nanometer. Then it undergoes simultaneously the electron transport system, wherein the ATP is produced from ADP and inorganic phosphate. The process is going to take place simultaneously, and the electron is further sent to one more light harvesting complex, the next light harvesting complex, and the same electrons they are utilized and they are sent to again electron acceptors. Here it is being reduced, NADP plus is then reduced to NADPH. Here we are utilizing the protons. Okay, NADPH plus H plus. Okay, the protons comes here. When NADPH reductase enzyme is there, so because of that enzyme, the process is going to happen, take place, wherein NADP plus is going to get reduced to NADPH in presence of protons. Okay, and cyclic photophosphorylation is something wherein the sunlight photon is being produced, falls on the chlorophyll molecule. This chlorophyll molecule, say again the electron will jump and it will be accepted by electron acceptor. This electron acceptor will then again further transfer the electrons to the ETS. Again, ETS will produce the ATP molecule, one ATP molecule. Again, the electron is sent back to the chlorophyll reaction center, light harvesting complex. Again, the same electron is being sent back to the electron acceptor. So this process continues. That's why this is called as cyclic photophosphorylation, where the process is a cyclic one and it repeats again and again. But here, in case of non-cyclic or Z scheme, why it was named as Z scheme? I hope everybody knows. Not the N scheme, but Z scheme, right? Now, this Z scheme or non-cyclic photophosphorylation here, the once when the electrons are utilized, again there should be photolysis of water so that the electrons again further they are carried. This is a non-cyclic photophosphorylation. For phosphorylation production of ATP molecules, photo means sunlight. So in presence of sunlight, if we are able to prepare the ATP molecules, then it is called as photophosphorylation. Remember that. Okay. So this is chemi-osmotic hypothesis, wherein we are speaking about one molecule of thalloid. This is the lumen of thalloid. This is inner membrane. This is outer membrane. Okay. The membrane, the membrane of thalloid. This is the thalloid membrane. So what happens here is in the lumen, there is production of proton. Everywhere from every process, the proton keeps on accumulating inside the lumen. Because of accumulation of lumen proton inside the lumen, there creates a concentration gradient. Concentration gradient is developed within the lumen. Okay, outside the lumen there is stroma. Means outside there is stroma. Inside the thalloid there is lumen. Khali space is called as lumen. Now here there is accumulation of a high amount of protons. Now which are the processes in which the accumulation of protons takes place? Let us see. One, there are three ways in which the proton accumulation takes place. One is photolysis of water, that is oxidation of water. You can also call it as oxidation of water, wherein we are forming the proton molecules. So because of this breakdown of water molecule into oxygen and protons and electrons. Electrons are somehow utilized by these electron acceptors, but proton gets accumulated in the lumen. So that's first method where the protons keep on accumulating in the lumen. The second method is reduction of plastophenol from plastophenol. So there are some intermediate compounds. So from the stroma, the electron acceptors are not present externally. So once the electron that gets excited from the membrane to the outer side, what happens externally there are no electron carriers. So again the electron has to come to the ground state. Now if you see here, electron is excited here. Electron gets excited here, but here at this point there is no electron carrier that can bring electron back into the membrane. So here there is a proton carriers which is present in the stroma, which we call it as plastophenol. It gets converted into plastophenol, and again that is how the electron is going to come down. Okay, and it is going to enter here, and when electron comes down, the proton from the stroma it gets inside the lumen. That's the second method how the proton gets accumulated inside the lumen, and again the electrons they are carried. Okay, and they are carried to the last again one more light harvesting complex. Again the electrons are taken outside pyridoxine reducing substance, and here on the outer side there is NADPH reductase, wherein the NADP plus H plus forms NADPH. Again this is taking place in the stroma, because of which again there is reduction of a proton in the stromal part. Because there is a reduction of proton in the stroma part, again there is a decrease in the proton contained in the stromal part, but there is increase of proton in the lumen part. Because of this difference that we observe inside the lumen, there is high proton gradient. In the stromal part, there is low proton gradient. Because of this, there creates a proton gradient difference, and because of which the protons will come out from the lumen by passing through ATP synthase, and structures that we refer here as CFO and CF1. These are chloroplast F0 particles and chloroplast F1 particles, kind of similar to F0 and F1 particles that we observe in mitochondria, but the location is little different. But the process is still the same. The protons will enter, will come out from this, and as a result, there is structural change with respect to ATP synthase as it rotates. And as it rotates, the ADP will combine with phosphate and forms ATP. Okay, so ATP is going to be formed. That's how the ATP synthesis takes place, and this is actually the electron transport system. Okay, so this is a three method wherein the energy is being synthesized. The next step is actually the C3 cycle, the main cycle, okay, during which the food is being prepared. Okay, C3 cycle, also called as Kelvin cycle, also called as Kelvin cycle. Okay, the first step here is carboxylation. Carboxy, matlab carbon dioxide. Lation, addition. So a carbon dioxide is getting added, but it is going to get added to which compound? Ribulose 1,5 bisphosphate. It's a 5 carbon compound. Remember this. Okay. Ribulose 1,5 bisphosphate is a 5 carbon compound to which one atmospheric carbon dioxide is added. This is carried out by Rubisco enzymes. Okay. What happens here is this addition of one more carbon to this ribulose 1,5 bisphosphate, it should create 6 molecules, right? 5 plus 1, I hope everybody knows maths. 5 plus 1 will result in 6, right? But no, we are not writing like 5 plus 1 is equal to 6. We are going to write it as 3 plus 3. Why? Because the 6 carbon molecule will split itself into 2 molecules of 3 carbon compound. Okay, those 2 molecules of 3 carbon compound is itself called as a phosphoglycerate. So phosphoglycerate is going to be formed. So this phosphoglycerate is going to get reduced. Phosphoglycerate is going to get reduced. Okay, forming triose phosphate. Now, during this process, whatever the ATP and NADPH was produced in the light reaction, okay, now this process is occurring the outside, right? Outside the stoma, I mean outside the gland, in the stromal region, C3 process is going on. As a C3 process is going on in the stromal part, in the stroma is itself they require ATP and NADPH. ATP and NADPH is utilized and 3-phosphoglycerate is then forming into triose phosphate. Okay. Now what happens in triose phosphate is a 3 carbon compound, right? Now out of 3 carbon compound, here there are total of 2 molecules, right? Among 2 molecules of 3 carbon compound, one carbon, okay, among all these 6 carbons, 1 carbon is going to come outside. So how many carbons we are left with is at all there are 6 carbons, and out of 6 carbons we are eliminating 1 carbon here. So we are left with only 5 carbons. 5 carbons in the form of 3 plus 2. So now this 3 plus 2 compound is again regenerated. For the regeneration of a 5 carbon compound, we again utilize ATP. So again we utilize ATP to form a complete 3 plus 2, that is 5 carbon compound. So that's how the ribulose 1, bisphosphate is formed. Okay, I hope this is clear. Again the process continues. So this is a 5 carbon compound which when added with atmospheric carbon dioxide, it forms again 2 molecules of 3 carbon compound. Again 6 molecules will be there, but among 6 molecules 1 carbon will get eliminated every single time. So this Kelvin cycle, it has to take place 6 times so that 1 molecule of glucose is formed. Okay, so 6 times carbon dioxide should enter into the C3 pathway so that 1 molecule of glucose is formed. And if you calculate here, uh, you know, ATP that is being utilized. Okay, one ATP there and one ATP here. Okay, but remember this is a 3 carbon compound, right? Which is going to take place 2 times. So one time here there is utilization of 2 ATP and here 1 ATP molecule because it is going to recombine, right? 3 plus 2. So it is going to recombine only once it is going to happen. So for carrying out one cycle we require total 3 ATP. Okay, and how many NADPH? 2 times. Obviously, this is going to happen 2 times, right? So NADPH for that is being utilized for carrying one cyclic reaction is 2 NADPH. So this is for one cycle. So how many times the cycle should run? The cycle should run for 6 times. 6 times the Kelvin cycle should take place. Now, here we have written 3 ATP for one cycle. So, how many ATP are required for carrying out 6 times? So, 6 into 3. A total of 18 ATP are required, right? And 2 NADPH are required in one cycle, right? So, 6 times if it should take place, 6 into 2, that is 12 NADPH. So if we put this input, we are going to get this output. So input is requirement of 6 molecules of carbon dioxide, 18 ATPs, 12 NADPH. When we add all these things, we are going to get 1 molecule of glucose. So to get 1 molecule of glucose, to prepare 1 molecule of glucose, the plant it requires 18 ATP and 12 NADPH. I hope this is clear. So 3 processes. Remember the sequence as well: carboxylation, reduction, regeneration. 3 steps in order. Remember that. Next, this is the C4 cycle, which we also call it as hatch and slack pathway. Then, here what happens here is mainly all the C3 plants. In C3 plants, what happens? The enzyme that was being used over there was Rubisco. Rubisco enzyme is very sensitive to it gets combined very easily with oxygen also as well as carbon dioxide. Now, if the amount of carbon dioxide is very less, Rubisco can combine with oxygen. If it combines with oxygen, the end product that we are going to get here, okay, the product that we are going to get here, yes, it's not mentioned here, the product that we are going to get is of no use because that is not neither going to yield any energy or neither the food is going to be formed. Okay, so that is what we call photorespiration. So usually the yield of C3 plants is less because of photorespiration. To overcome that photorespiration problem, when the Rubisco should only combine with carbon dioxide, not oxygen, here the some plants, okay, some plants showed some changes. Those plants are itself called as C4 plants. So in C4 plants, there are 2 different cells. One is mesophyll cell, the other is bundle sheath cell. Mesophyll cells are in contact with the atmospheric carbon dioxide. So here the Rubisco is present in the bundle sheath cell. It is present inside. So when it is present inside, it cannot directly come in contact with the atmospheric carbon dioxide. Even if oxygen is present, it is not utilized by mesophyll cells. So there is no waste of energy, right? So therefore, the atmospheric carbon dioxide is utilized by bicarbonate ion. This bicarbonate ion, when combined with phosphoenol pyruvate, it forms a C4 acid. There is a C4 acid which we'll learn later in the Krebs cycle. Okay, there is a C4 acid. Now this C4 acid is getting converted into another C4 acid and forms the C4 acid inside the bundle sheath cell. Okay, this conversion is very important. Fixation and conversion. Phosphoenol pyruvate is a 3 carbon compound. To this 3 carbon compound, there is addition of 1 more carbon dioxide. So that carbon dioxide when combines with the C3 compound, it forms a C4 compound. That C4 compound is transported into the bundle sheet cell, which we call C4 acid. In C4 acid, for this C4 acid, there is a process called as decarboxylation. Carboxylation we had learned it in previous cycle itself, C3 cycle. Carboxylation means addition of carbon dioxide. Decarboxylation means removal of carbon dioxide. So we are removing the carbon dioxide, and that carbon dioxide is given for the process of C3 cycle. Again, the same C3 cycle. Just now we have learned the same C3 cycle runs in the C4 plants. Okay, but inside bundle sheet cell. So we should remember that inside bundle sheet cell there is a C3 cycle that is taking place. Okay. Now, if from C4 compound, it's very difficult question. From a 4 carbon compound, if you remove one carbon, how many carbons will be there? Only one carbon will be eliminated. A total of 3 carbons will be there. So 3 carbon is present here. The same 3 carbon is converted into one more form. Again, that is sent back to mesophyll cell. In mesophyll cells, what happens? There is again a regeneration of a phosphoenol conjugate. So again, P is formed. Okay, I hope here it is clear. Okay, and this is one of the factors affecting the photosynthetic process. Light, temperature, water, sunlight, carbon dioxide concentration. All of these factors affect the rate of a photosynthesis. And the one law that is being given in your textbook is Blackman Blackman's law of limiting factor or limiting reagent. Limiting reagent, which you have also learned in chemistry, the same process. Among the four factors, light, so that's sunlight, temperature, and water, as well as the concentration of carbon dioxide. Among these four, the major limiting factor is actually carbon dioxide. Okay? If carbon dioxide goes beyond the limit of minimum requirement of the plants cannot carry out the process of photosynthesis. Okay, I hope it is clear. So this completes the 11th chapter. We are moving to the 12th chapter, that is respiration in plants. So the respiration of plants, the main uh, first very important process that we see is glycolysis. What is glycolysis? The conversion of glucose into pyruvic acid is called glycolysis. Okay. So glucose. How actually this glucose is formed? From sucrose. Okay. An enzyme called as invertase is used to convert one molecule of glucose and one molecule of fructose. So one molecule of fructose and one molecule of glucose is formed. This glucose, which is a 6 carbon compound, we are going to add one phosphate group. Okay, on the sixth position, that is glucose 6 phosphate. Okay, now for that to occur, we are going to add nitrokinase enzyme. Okay, now this glucose 6 phosphate is in turn converted into fructose 6 phosphate. Okay, so there are numerous tricks to remember, but instead of remembering the tricks, try to revise the concept, make your concept strong. What happens when you use the trick is you cannot go on remembering that was something, that was a trick which I forgot. In the exam, you cannot afford to use even a single minute also. Okay, NEET is not an exam wherein you score 200 and get a medical seat. Okay, scoring 200, 300, 400 is not something what you have to aim for. If you are giving medical exam, you have to aim for scoring more than 550, 600. That should be your primary target. Okay, so for scoring that, your concept should be very strong. Okay, then so glucose 6 phosphate, fructose 6 phosphate, fructose 1,6 bis phosphate. Then this fructose 1,6 phosphate is broken down into two different forms. One is glyceraldehyde 3 phosphate. That is, in short, we call it as glyceraldehyde 3 phosphate, and dihydroxyacetone phosphate. That is GHUP. Okay, so again they are interconvertible. One molecule of glyceraldehyde 3-phosphate takes part and it also forms 1,3 bisphosphoglycerate, 3 phosphoglyceric acid, 2 phosphoglyceric acid, phosphoenol pyruvate and pyruvic acid. Now that is one time. Again in this dihydroxy acetone phosphate, it also gets converted into glyceraldehyde 3-phosphate and one more time the same process happens. So totally we get at the end we are going to get two molecules of pyruvic acid. At the two places ATP is utilized. At two places ATP is generated. Okay, and at one place NADP is also NADH is also generated. We have to see this. Okay, first and third. Remember, first and third both are very dangerous. Remember, if you come first, next time also you have to come first. That will be there in your mind. If you come third, you start thinking that what if I don't come second, or what if I don't even come third? Second is good, at least they can come first and third. But first place, they feel fear that if we lose first, what happens? So first and third are very dangerous. Remember like that. So first process in the first process, ATP is utilized, and in the third process, here also the ATP is utilized. So the places, the sites or the steps in which the ATP is utilized is the first and the third step. Remember that. Then ATP is produced, okay, in the last step and the last to fourth step, okay, which we commonly call it as first, second, third, fourth, fifth, sixth step, and ninth step. So sixth and ninth, okay, multiples of 3. You can remember 6 and 9. That's where the ATP is produced. Okay, how many times the ATP is produced here? Okay, 2 times the ATP is produced. So that means 2 into 2 ATP. That will be 4 ATP. 4 ATP are produced. And one molecule of NADH is produced, and that is also an energy rich compound. So how many ATP NADH molecules are produced? 2 NADH are produced. Okay, 2 NADH are produced. But 2 molecules are of ATP are already utilized. So how many net ATP are remaining? 2 ATP are remaining. So the total gain. So the total net gain, if we have to talk about total net gain of glycolysis, 2 ATP are present and 2 NADH are present. Okay, remember that. Net gain is 2 plus 2. Whereas the overall, the irrespective of whether it is net gain or how many amount of ATP are produced, you should always remember that 4 ATP and 2 NADH are produced, but 2 ATP are utilized. The net gain of glycolysis is 2 molecules of ATP and 2 molecules of NADH. Okay, that's the most important part here. Okay, that's regarding glycolysis. Okay, then so this is a the fermentation process wherein there is alcoholic fermentation as well as there is anaerobic fermentation. Glycolysis is something which occurs irrespective of whether it is a prokaryotic cell or eukaryotic cell. In both prokaryotes and eukaryotes, the glycolysis occurs irrespective of whether they are aerobic or anaerobic. The most important point is this: irrespective of whether the cell is aerobic or anaerobic, the process occurs. Okay, the glycolysis occurs in the cytoplasm of both aerobic as well as anaerobic organisms. Remember that. Okay. Now, in case of aerobic organisms, the further process is Krebs cycle, aerobic. But in case of anaerobic organisms, the pyruvic acid, pyruvic acid can be either converted into lactic acid or it can be converted into ethanol. Lactic acid, okay, is usually produced in bacteria, or if we have to speak about human beings, in the muscles of human beings. If oxygen is less, so if there is less amount of oxygen, if you do more work, what happens? The lactic acid gets deposited, okay, in the muscles. Now, again, for the conversion of pyruvic acid into lactic acid, there is a utilization of one molecule of NADH. So, how many pyruvic acid are formed at the end of glycolysis? Two molecules of pyruvic acid are formed. So, again, this process takes place twice. So, two lactic acid are formed. So, two lactic acid are formed, and two molecules of NADH are used. The whatever the net gain of glycolysis was there, that is 2 ATP and 2 NADH. Okay, 2 NADH are utilized here only. So the remaining energy that will be there for bacteria is just 2 ATP molecules from one molecule of glucose. So from one molecule of glucose, bacteria are getting only 2 ATPs. Okay, which is very less, which is less than 7% of the total energy that one glucose molecule can give. Now, even if we talk about the conversion of pyruvic acid into ethanol, this is taking place in yeast. So, yeast, what is happening in yeast? If you remember, it is used for breeding. It's called as breeder's yeast because when we add yeast, the ethanol is going to be formed. Alcohol is going to be formed. Therefore, this process is itself called as alcoholic fermentation. And in the alcoholic fermentation also, there is formation of two molecules of ethanol and utilization of 2 NADH. Again, 2 NADH is utilized. So, how much of the energy will be left with the yeast? Again, just 2 ATP molecules only. So, all these are anaerobic processes. If they take place, then what is going to happen? Either there is alcoholic fermentation or lactic acid fermentation. Okay, lactic acid fermentation takes place in bacteria. The energy that they will be left with is only two molecules of ATP, and here again in yeast also we see two molecules of ATP. Then, as we discussed, remember when we were discussing biomolecules, I said magnesium ion plays a very important role in link reaction. This is a link reaction. Link reaction in aerobic organisms. Now we are speak only about aerobic organisms. So before entering into this, one complete glycolysis process yielded one molecule of glucose gave two molecules of pyruvic acid. Two molecules of pyruvic acid was there. Okay, and here this pyruvic acid we should convert it into acetyl coenzyme. Okay, pyruvic acid must be converted into acetyl coenzyme A. Now, to convert it into acetyl coenzyme A, we require coenzyme A. Okay, we require a cofactor that is coenzyme A and NAD plus. NAD plus. The enzyme that we are using is pyruvate dehydrogenase. By utilizing pyruvate dehydrogenase in presence of coenzyme A and NAD plus, pyruvic acid is going to get converted into acetyl coenzyme A, and carbon dioxide is released. Carbon dioxide is released and NADH is produced. So here there is no utilization of energy, but actually there is formation of energy molecule here. Okay. Here, two pyruvic acid, all these processes are going to occur two times. So, at the end, we are going to obtain two molecules of acetyl coenzyme A and the most important two molecules of NADH. Okay. Earlier in glycolysis process, we already had 2 ATP plus 2 NADH. Now we are again getting 2 more molecules of NADH from just a link reaction. Then we are entering into the Krebs cycle. Very important point here. Okay. Now, how you should remember this? Here, there is a 4 carbon compound, that is oxalic acid, oxaloacetic acid. Okay, we commonly call it as OA, OA. This is link reaction. Just now which we discussed. Pyruvic acid requires one molecule of coenzyme A, one molecule of NAD plus, and in presence of magnesium and pyruvate dehydrogenase, it is going to form acetyl coenzyme A. Acetyl coenzyme, and it is going to pure carbon dioxide, because pyruvate is a 3 carbon compound, acetyl coenzyme is a 2 carbon compound, one molecule of carbon dioxide is getting eliminated. Okay, now this is OA. Can we just remember this as okay? OA and acetyl CoA. Can we remember it as acetyl CoA? Just for your understanding purpose, OA when combines with CoA. Understand that OA is a friend. It is going to call its another friend, that is acetyl CoA. They both are very good friends. OA and acetyl CoA. OA is calling acetyl CoA, and they both sit together. 4C plus 2C, they both are going to sit together, forms 6C. Remember, OA sits with OA, they both will sit together, forms 6C. OA had brought oranges for acetyl CoA. That's why we call it as citric acid. Remember, citric acid is a very important acid present in oranges, right? So, OA had brought oranges for acetyl CoA. So, that orange is having the citric acid, they both will sit together, forms 6 carbon, and the citric acid is formed. Now, both of them, they will be like very good best friends. Their friendship is something like, it is very strong friendship. It is very strong friendship that it's like glue like. They both are bound very strongly. Alpha keto glutaric acid, glutaric acid. Their friendship is glue like glutaric acid. Okay. As they eat orange, one orange. So one orange is going to get eliminated. CO2 is going to get eliminated here. Okay. Now, if the CO2 is getting eliminated, how much carbon will the alpha ketoglucaric acid will have? That is okay 5 carbon. Okay, it is going to have the 5 carbon. Then the alpha ketoglucaric acid they both will sit and study very hard. If they sit and study very hard, definitely they are going to get success. That is what we call succinic acid. And also they are going to eat one more orange, means one more carbon dioxide is getting eliminated. So succinic acid is a 4 carbon compound. Remember that. Okay. And then succinic acid is then converted into malic acid. Malic acid is then again converted into oxaloacetic acid. Okay. Now that's one part. That's one part of remembering all the things here. But how to remember the energy that is being produced? Just divide this part into half. The link part you remember there only. Don't go to remember from here. If you remember, no worries. This from half uppermost part, there is utilization of or formation of NADH. Okay. Again, in the lower part, again there is formation of NADH. Again, the NADH is formed. Here in the uppermost part, again there is formation of NADH. But here there is formation of FADH2. Now, to manage this FADH2, here just we are going to write it as a GDP. Okay, so how many NADH molecules are formed from one pyruvic acid? One pyruvic, total of two pyruvic acid are there from one pyruvic acid. From one pyruvic acid. From one pyruvic acid, if we include a linked reaction, again there is formation of two one NADH molecule. Okay, initially in the linked reaction we discussed one NADH molecule in the linked reaction. Then here 1, 2, 3. Okay, in the Krebs cycle there is formation of 3 NADH molecule, and again in the Krebs cycle only there is formation of FADH2 plus one molecule of GT. So overall of all these are utilized, okay, in for the from the one pyruvic acid. So if we take a two pyruvic acid, so double this, okay, 4 NADPH are there, 8 NADH, okay, plus discard 2 is 2 FADH2 plus 2 GDP is nothing but another energy currency which is similar to ATP. Okay, so that is the total energy that we get from one molecule of glucose. Okay, in the Krebs cycle and lead reaction part. Now, if you just see, that is what they have given. One molecule of pyruvic acid. One molecule of pyruvic acid yields 4 NADH molecules, one molecule of FADH, and one molecule of ATP. ATP is nothing but they are talking about GDP. Okay. Now, as you know, from glycolysis, there is always formation of 2 molecules of pyruvic acid. So this when we double, we are going to get 8 molecules of NADH, 2 molecules of FADH2, and 2 molecules of GDP or ATP we are going to get. All this process is going to take place in the mitochondrial matrix. So remember the matrix. There we are going to see all this process. Okay. Then this is ATS process. So remember the complex thing. That's what you should remember here. Okay. Complex one is NADH dehydrogenase, wherein the NADH is going to get reduced to NAD. Again, here the electron transfer takes place. Ubiquinol. In the cytochrome 2, I mean the complex 2 molecule. In complex 2 molecule, the succinic acid, okay, is getting converted into fumarate. Again, here that is nothing but FADH2 is getting converted into FADH, okay, FAD. So FAD is going to be formed. Again, the electron transfer takes place from ubiquinol to ubiquinol, and the electrons are going to come to cytochrome BC complex. Okay, remember, complex 1 is NADH dehydrogenase. So यहाँ पे there is the removal of hydrogen from NADH compound. So NADH dehydrogenase. We are obtaining two compounds, right? One is NADH and FADH2. Now FADH2 say we should remove H, again hydrogen, and NADH say again we should remove hydrogen. So in the complex one we are removing hydrogen from NADH. So just remember it as complex one. NADH. What we are removing? Hydrogen. So that's what we are going to call it as dehydrogenase, removal of hydrogen. Very simple. Then in complex 2, second complex, we should remove hydrogen again from FADH2. So how we will remember FADH2? Remember it as succinic acid. Succinic acid से क्या remove कर रहे हैं? Hydrogen. So therefore we are removing hydrogen from succinic acid, that is nothing but FADH2. From FADH2 we are removing the hydrogen, so that's why we say it as succinic dehydrogenase. Okay, so that's how the complex 2 name goes. That is succinic or succinic dehydrogenase. Now, from both of these, the hydrogen, whatever we are removing, the proton that we are removing, it is going to get accumulated in the intermembrane space. Remember, we discussed intermembrane space is the space between outer membrane and inner membrane of mitochondria. That is the place where the proton gets accumulated. Okay, and the complex 3 is cytochrome B6. Okay, wherein the again electron is being passed. Complex 4 is cytochrome C oxidase, and finally complex 1 is the ATPase, wherein the proton gradient is developed here. If you already see here, matrix, from a matrix the proton is going to get accumulated more in the intermembrane space, similar to the chloroplast. You shall see inside the human the proton gradient is developed. Here inside the intermembrane space there is a more proton. Okay, to break that gradient, the proton from the fifth complex will enter into F0 initially and then F1 particle and comes out. So the protons goes on coming out. As a result, there is rotational conformation change. There is a change in the F1 particle. Okay, it rotates. As it rotates, the ADP, in presence of inorganic phosphate, it is going to form one molecule of ADP. Okay, so that is how the ETS works, and this is the ETS. Okay, try to just remember where what is going to happen. Okay, the intermembrane space is the place where the proton gets accumulated. Matrix is the place from which the proton gets released. Complex 1 say you are removing the hydrogen from NADH, and complex 2 say you are removing from FADH, but you just have to say it as succinate dehydrogenase, and complex 3 is cytochrome BC complex. BC comes in the middle, so you have to remember like that A, B, and BC comes in the middle. So then in the fourth complex that is cytochrome C oxidase. Okay, remember like that. Then so this is the all the pathways. Okay, even if there is a fat will then get oxidized to form fatty acids and glycerol. Fatty acids will can directly form acetyl coenzyme and vice versa. Okay, they are interconvertible fatty acids and acetyl coenzyme. Glycerol is then converted into DAP. Remember glycolysis process DAP and DAP. So glycerol can form dihydroxyacetone phosphate, which is in the interconvertible glyceraldehyde 3 phosphate. This glyceraldehyde 3 phosphate again completes the glycolysis process and forms pyruvic acid. This pyruvic acid then converts into acetyl coenzyme, which is going to take part in Krebs cycle. Okay, and ultimately Krebs cycle will yield the energy compounds. Okay, then not only that, this is direct glycolysis process. Again, from protons say amino acids are formed. Amino acids can form pyruvic acid. So again, the process continues. This is very important. Okay, it has been asked in the previous year also. Respiratory question. What is respiratory question? Don't get confused between which is numerator and which is denominator. The carbon dioxide that is evolved by consuming oxygen. इतना ही है. Remember this much only. Okay. How much amount of carbon dioxide is being evolved for the oxygen that is consumed? That is respiratory quotient. Okay. What is respiratory quotient? Respiratory quotient is nothing but the amount of carbon dioxide evolved by the volume of oxygen consumed. is respiratory quotient. Now C6H12O6, okay, it is oxidized completely. Okay, partial oxidation is glycolysis, but complete oxidation when we do, we require 6 molecules of oxygen, and 6 molecules of oxygen means a total of 6 O2 are required, and at the end we are going to get 6 CO2. So 6 CO2 divided by 6 O2. You don't have to cancel O2, 6 O2, and you should not write this C is what we are going to get, not like that. Okay, don't cancel in that. You should cancel complete 6 molecules of carbon dioxide and 6 molecules of oxygen. That is, the respiratory quotient is always 1 for carbohydrates. Remember only this for carbohydrates. For carbohydrates, the respiratory quotient is always 1. Okay, and this is a tripalmitin, which is a fatty acid. For fats, the respiratory quotient is 0.7. Simple trick you can use only the simple math. Okay, RQ, RQ values. Okay, RQ values. For carbohydrates, you have to remember for carbohydrates, it is 1. For fats, okay, whatever the F is there, do ulta of F. How you will write? Like this, right? So just put point here. So for fats, the value is usually zero point 0.7. Not only that, again for proteins, the value, how to identify? P, right? Ulta, mirror image of P. It is somewhat like this. That is nothing but 9. Zero point 0.9 is the value for proteins. And for organic acids, it is more than 1. This is what you should remember in the respiratory quotient. And this is the part of the last part of the respiration in plants. Next is the plant growth and development. In the plant growth and development, we see the first how the germination and seed development takes place. From hypo refers to lower part, cotyle, the cotyledon. Okay, the lower part of the cotyle forms the lowermost part, and epicotyle or the topmost layer, it forms the shoot system. Okay, and how the roots are formed from which part? The radical. The shoot is formed from lumen. Remember that. Then you know how the tip part, the shoot cut tip part is called as shoot apical meristem. The root cut tip part is called as root apical meristem. Okay. There is presence of vascular cambium in the middle that shows secondary growth. Okay. The how slowly the elongation takes place between the different parts. Okay. Initially it will be of this length. Slowly as the plant grows, there is elongation of cells. Okay. We see different graphical representation, diagrammatic representation of growth. If we see arithmetic growth, what happens here is only one cell keeps on dividing, the other cell will not divide, they lose their ability to divide. That type of division or growth that we observe is an arithmetic division. Okay, what happens in arithmetic division? It's a type of division of cells in which only one cell will retain the property of division. Okay, again the next time only that cell will divide. Again the next time only that cell will divide. So only these side cells will divide. So this side cells cannot divide. Okay, and in geometric division or geometric representation, all the cells they have the ability of undergoing division. Then the third one is kind of you know what we call it as both geometric as well as arithmetic. It's a mixture of both arithmetic as well as geometric division. Zygote is having the ability to divide. The initially zygote will form two cells. Zygote is unicellular. But zygote when divides, it forms two cells. Two cells will again divide, forming four cells. 4, 8, 8, then 16 till here. Okay, all the cells they have the ability to again undergo division. But after it reaching particular phase, again only some cells will show the property of cell division, whereas the other cells they start becoming permanent. Okay, that's again the growth how it takes place. Just add this in. So this is how the growth is observed. In some plants, the relative growth means the growth of one particular uh, component with respect to other is called as relative growth. Now, if you see the comparison between this both A leaf and B leaf, A was the initial measurement of the leaf. A dash is measurement after a period of time. B is the original length of the leaf, whereas area of the leaf, whereas B dash is the later measured area of the leaf. Now, when you compare A and B, you can say that in the same period of time, A shows a higher growth rate because the earlier area was just a 5 centimeters square. Then within some period of time, this 5 centimeter square became a 10 centimeter square. So almost the size was increased 50%. Yes. So now when we come here, okay, when we come to this point, what happens here is the earlier measured area was 50 centimeter square. Now as it was 50 centimeter square, after a certain period of time, it became 55 centimeters square. So the increase in the area was around only 10%. Now how to remove that? 5 point if we give, it becomes 10%. So 55. So only there was increase in the 10%. So is 50% greater growth rate or 10% growth rate? It is more. So obviously the first part shows higher growth rate when compared to the second leaf. Okay. So that is how we have to see or observe the changes. Okay, a very important part here. Okay, every time the niche, okay, India people they have a very keen eye on this concept. Okay, of asking differentiation, re-differentiation, and de-differentiation. Okay, that's a very important part. Okay, which I will tell initially. I guess it is not mentioned here. One second. So what we will do here is, okay, when the plant is meristematic, it is not having any specific role. So this meristematic tissues will then differentiate. Very important for the first time, they will undergo differentiation, or they will differentiate. They will differentiate to form permanent cells. They will differentiate to form permanent cells. Now, these permanent cells have lost their ability of undergoing division. But if you remember, we said about secondary growth. Some cells they show secondary growth in case of dicots and gymnosperms. So there are also permanent cells on it. They are actually parenchyma cells. But after reaching a certain point, these parenchyma cells or the permanent cells, they will again gain the ability of becoming meristematic cells. They again become meristematic in nature. So that ability of permanent cell becoming meristematic is a redifferentiation. And these meristematic tissues again they will divide and they will again form permanent tissues. These permanent tissues for the next time when they divide, that is what we call differentiation. So remember that. Okay. Differentiation is when meristematic cells first time they divide and form the permanent cells. The permanent cells, when they become meristematic in nature, that is what we call redifferentiation. The meristematic cells again, once when they de-differentiate and form the permanent tissues, that is what we are going to call it as de-differentiation. Okay, so the plasticity, the one more ecophyty. Okay, if it is present in terrestrial habitat, the arrangement of leaves is different. If it is present in water habitat, the structure of leaf is different. So depending on the environmental conditions, the same plant. Okay, the A is meant for larkspur and B is meant for buttercup species. The same species, but when they are present in different environment, they show different type of leaf arrangements or the units. Okay, that we also call it as plasticity. Okay, the same the tip of the oxygen it was used to extract the tip of the rice was used to cut and to obtain the oxygen component. Okay, first time it was observed by Spook. So they cut the tip part and they observed that wherever the light was coming up in the direction of light, the tip was bending. So that is where they actually found that oxygen was very sensitive to the light direction. Okay, and then again apical dominance. The tip part is dominant, and if the plant continues to grow straight, what happens is they will cut the tip part. If they cut the tip part, slowly the plant starts to grow, you know, uh, laterally as well. If the tip part is cut, the plant will start growing laterally to just to reduce the apical dominance. Okay, so that completely ends the plant physiology for our part. We are going to take a very short break here. We will be back at 7:30. I'll just upload these files and you can observe it as well. So, wishing you all the very best for tomorrow's exam and give your best 100%. Go through the video and just revise week up. Okay, no need to panic if you don't remember. Try and utilize your current time for revising and just relax. No need to do new concept, no need to by hand things. Whatever you have done your best. Revise, recall, and then give your exam tomorrow. Okay. And the only remaining part is regarding the physiology part here, which I think so maybe did not possibly have. Again, as you people require time for reading as well. So better you take your time, utilize your time for your preparation. If you have any doubts, you can just put a comment in the comment section. I will address your questions as well. Okay. So thank you everyone, and all the very best for your NEET examination. I hope you all are most of you, okay, who have done the hard work will definitely achieve success in this tomorrow's NEET examination. Don't get panicked, and all the very best for tomorrow's examination. Thank you. This is BioMedic Guruji signing off for today.